Hello and welcome to today's episode of the Out of the Lab podcast. Today I'm joined with Jennifer, VP at Catalent. Jennifer, welcome to the show.
Thank you, Nicholas. Thank you for having me.
So Jennifer, the first question I'd love to get into on today's sort of technical episode is what trends you see in terms of missing details in incoming procedures from your vantage point. Is it missing details, vague qualitative language, or something else? This is one of the big themes on the show.
Yeah, for sure. I think it's understandable that where the clients are coming from is they are very early stage, right? So the majority of customers that we find coming into our lab initially, they've got an idea. They might have a few clones selected that they're trying to narrow down, but they don't know a lot yet about their product or about the methods that they need to use to measure their product. They're trying to figure out their key quality attributes, right? And that's all completely understandable. What that comes with, though, is sometimes documentation that is incomplete and incomplete from the perspective of a GMP lab and necessary to run in a GMP laboratory in a GMP environment, but complete enough to run the assay or to do what we need to get a result. To answer your question, though, where we're really seeing the trend in the missing information, it's around the specificity of the step. So a lot of assumptions that if you are, a scientist working on an early stage project and you're just trying to get something moved forward, you might not consider just because it's not been for you to consider before. It's not been something you've had to consider before.
Right. Yeah, and that sort of makes sense. Now, are a lot of these things things that you might expect someone to take for granted if they're working in the lab, details that might impact a procedure that because they're sort of in the background environment, you might not sort of consider, or are they really part of the protocol from your perspective?
Yeah, I mean, I almost think it's one and the same. I think, every individual probably cuts their steak differently, or cuts. I should say
Yeah.
vegetable because not. So every individual cuts their vegetables differently, and you don't really pay attention to how you do this. But if you take that analogy and you take it into the lab, everyone, probably calibrates their pipettes a little bit differently. Do you do it on a 3-month cycle or a 6-month cycle? How rigorous are you around just even the calibration of your pipettes? If you're in a research lab, probably less so than if you're in a GMP laboratory, right? But even beyond that, assuming that the pipettes that you're using and the plates that you're using are like for like, the technique is going to vary just because we're all different people. So there is going to be human variability that's introduced. So when we're talking about assumptions that are being made, it's natural because again, it's not been something that anybody has had to try to control before for this particular product or this particular assay. And that's where the connection with a GMP scientist or scientist that has worked in a GMP laboratory becomes really useful because we have seen it before. We can point out kind of where the specificity makes maybe more of a difference than not.
That makes a lot of sense. So one of the one of the words that's come up a lot during these conversations is gently, like gently resuspend, for example. And I'm curious, do you see more of these problems coming from the sort of first you discussed, which was more like quantitative attributes like pipette calibration, things that are done on a schedule, as opposed to trying to deconvolute what someone meant when they said gently resuspend, for example, or like how they might have stirred a flask, something like that.
Yeah, definitely the latter situation. I mean, again, it's that human variability that we're attempting to control. And so you don't know what you don't know until you put a bunch of humans on the same project, and then you start to see where the variability comes. And that's an exactly perfect example. Gently resuspend. What does that
Yeah. And, I'm curious from the GMP perspective, is there, I know that often someone will be sent, a scientist from the sending lab. Is that the current best, what is the best practice right now for resolving these types of, misunderstandings or, yeah.
Yeah, I think it's a good question. I see labs coming at it a various different ways. So some labs, and I'll say the lab that I'm working with now, have very stringent techniques that we apply to every incoming method. So we know what decreases human variability. So we would change language with the customer's input, of course, to move from gently resuspend to, maybe invert the tube three or well, I shouldn't say 3 or 4 times, but invert the tube 3 times, whatever it is. But we would go through and help the customer specify the language based on our techniques and what we have found to reduce human variability. I'd say that's probably the best approach, honestly, because that is theoretically scalable, or at least you can apply that learning to other assays, right? Now, we will, even with that method and that approach, we will get into some situations where we are unable to control the variability and it becomes unclear as to why. And in that case, working scientist to scientist makes a lot of sense. And so in some cases, we'll have the scientists come to our lab in Kansas City, and in other cases, we'll send scientists to their lab, but essentially watching what each other are doing to hopefully identify where that source of variability is coming from. I hesitate to go to that for, the first step, only because it's not very practical to do that for every single assay that we see. So we do try to decrease the variability with techniques that we have seen work in the past. And then if that is not enough, then we would do definitely the scientist to scientist connection.
Okay, so sort of like a first pass, you might look at a protocol, identify things that might be unclear. Hopefully that will resolve it. And then if not, then it would actually come to sending someone to another site.
Yeah.
Okay. And I'm curious. I think with Catalent, there's like a variety of modalities there. With cell and gene therapy, it's a very new modality, and one of the consequences is there's a lot of open processes. Not as much equipment has been designed as, for example, vaccine manufacturing. And I wonder if you see differences in these like really cutting edge technologies that, a lot of the time we're talking about replicating what a research scientist did um, at a sort of larger scale. Do you see that sort of impacting the like the likelihood that you need to send someone back and forth, or that you might have these kind of challenges?
Yeah, that's an interesting question. Yes, in some situations, I would say. I think so cell therapy, certainly every assay is pretty de novo. And so the ability to work with that assay and that product type is unique to almost every product. So in that case, obviously there are best practices that we can apply across the board. But the likelihood of needing that kind of technical to technical discussion is uh, experimentation is definitely necessary. Gene therapy, ironically, we're seeing more and more almost platform type approaches. So we're not seeing quite as much variability in gene therapy, which kind of makes sense when you look at cell therapy versus gene therapy anyway. Um, we do see, and this sort of surprised me when I started looking at the trends, is that antibodies have been around for a long time. However, what people are doing with them now, with bispecifics and trispecifics and conjugation and ADCs, is very different. So we're actually finding that is prompting some closer technical discussions than we've needed to have previously. So we can no longer take for granted just because it's an antibody, we know what to do with it, which is cool. I mean, that's part of, one of the reasons I love working in this industry is there's so much innovation going on, even with things that seem like they've been figured out in the past.
That is, yeah, that's really interesting to hear that. So you mentioned about the platform technologies. That's sort of the question I have about that is, um, do you think that that reduced variability when you do have platform technologies, um, is that because you might be more comfortable with that specific step? So if you have, a first customer who's used a platform and you get to the second, third, and you're like, okay, we're very comfortable with this now. That is different from when you're completely rebuilding your own stack as a new customer.
Yeah, I think absolutely. And I think this is where we see the trends going, in with our customers, especially I was going to say larger customers, but emerging biotech as well, right? It makes sense to do something really well once and then make a change and repeat it as close to that initial exercise as possible. It's much faster, obviously costs a lot less money. But then, yeah, if you are working with the same lab through that to also, as you bring more molecules into your pipeline in a platform approach, and we've got the platform assays that we become more and more familiar with, that we are trending where the problems are over time. We can catch them very quickly if there are problems, but ideally, we are starting at a place that is a bit more stable to begin with, right? That we understand the method and the challenges with the method.
Yeah, yeah, that makes a lot of sense. So you mentioned there's sort of a difference between gene and cell therapies in this regard. What are your thoughts on that? Like, is there, is it like a technical? Yeah, where do you think that's coming from?
Um, I mean, primarily it's the sample type. I think as an industry, there's less gene therapy material to work with for assays than there is for antibodies, right? And there's even less than that for cell therapy. Not to mention that the cell therapy is very sensitive and needs to be handled with care. In some cases, these products don't have a very long shelf life. So it's just the sensitivity and the limitation really of the material.
Yeah, I mean, yeah, that makes a lot of sense. Now, as we're sort of coming towards the end, what do you see as like sort of the future direction? For example, maybe you have some thoughts towards future scientists that might be approaching maybe their first tech transfer. Yeah, what suggestions might you have as they might be wrapping up their protocols and thinking about transferring into a GMP environment?
Yeah. Number one would be raw materials. Where are you getting your raw materials from? And are they single sourced? Ideally, you would have at least, and this is not possible, I know, in some cases, but ideally, you would have at least two sources for the critical raw materials that you will have tested in your assay, so that if there is a supply challenge with one, especially given our current environment, that you can easily move to a second supplier. So I would say that's number one. And then the second thing would just, be a scientist, have fun with it, find the limitations of your assay. Ask yourself, okay, this worked. I did this whole step for 5 minutes. What would happen if I did it for 10 minutes? Um, and I would also layer that with keep in mind, think about who's going to be running this assay long term. Usually, these are scientists that work in a lab for 8 hours a day, Monday through Friday. So, if we have a postdoc that's working on a really cool assay that they're willing to stay up for 48 hours, well, that is going to cost a lot of money to run in a traditional GMP laboratory to staff for 48 hours. So kind of to think about those things as you're designing your assay and just have fun with it, test the limits of it, and make sure that you write those down so that you can transfer that knowledge to your service provider.
Yeah, that's a great piece of advice, and yeah, timing the metabolism of cells in terms of work days can certainly be an ongoing challenge.
It's fair, and we come in on the weekends, and we have shifts that can handle that variability, but we also try not to overload the workforce with unrealistic timeframes where it can be avoided.
Awesome. Well, thank you so much for joining us on today's episode.
All right, thank you, Nick.